name is Shihan. Today I will present the topic 2 assignment. In this workshop, we practice the chip-seq data analysis. We will combine the concept of gene expression and epigenetic, learn in this topic and analyzing the interaction of three transcription factors in S288C strains during gene expression under osmotic stress and regulatory change of gene expression. Chip-seq is most commonly used to study DNA binding proteins such as transcription factors, chromatin-modifying enzymes, modified histone that interact with genomatic DNA, and the components of the basic transcriptional machinery. In our workshop, we will use chip-seq to focus on analyzing the part of gene expression. The datasets we used for this workshop was obtained from the research experiment done by Kristen E. Cook and others. The targets we want to study are three transcription factors, which are HOG1P, SCOL1P, and HOT1P. These three factors are all from the same strand. From Uniport, I summarize the biological function of these genes as left table shows. We separate the chip-seq dataset into two groups. The control group, the cell were growing at 30 degrees in YEPD with shaking. The treatment group, same growing conditions but added osmotic stress. For our cases, we're using FastQC to do the quality check for the sequences. From an overall result, we can see that most sequences data has a good quality, except one mock sequence data. We use the bowtie tool as a sequence alignment tool, then use SAM tools to filter and sort aligned sequences data. Use MAX tool to recognize the peaks in the sequences, then use chip-seeker to do the peak annotations. But before the annotations, we need to change the ref-seq in the MAX tool output file to chromosome identification to meet the needs of the annotation process. We use digest to calculate the expression log fold change for the RNA-seq data. From the result, it shows the mean precipitate is the promoter, but there are also a small number of exons and other material. Most promoter are size less or equal to 1 kb. The HOT1P shows a unique result in which the peaks from the KCI result are only promoters less or equal to 1 kb. The YEPD contains no peaks. This distribution shows promoter less or equal to 1 kb are the main components of TSS, which will facilitate the binding of transcription factors. I filter the RNA sequence expression result with the gene ID from the annotated peaks to retain the relevant results. This is to show how genes from the peaks are expressed over different time points. I have drawn the heat maps according to each annotated peaks result. HOT1P does not have any relevant gene ID with RNA sequence data, so no heat map can be drawn. From the journal, the list indicates that the gene ID represents the ORF of the gene, so I build an index according to the list to rename the genes for the heat maps. Combine the top 10 expressed genes with the biological processes. We can see the most expressed genes under YEPD condition for HOG1P are related to cell metabolism functions, and the most expressed genes under YEPD condition for scope 1 are related to cell metabolism function as well. The most expressed genes under KCI conditions for HOG1P are most related to stress response. We can also see a similar result for scope 1 under KCI conditions. Comparing the expression profile of HOG1, we can see that due to osmotic stress, some genes are removed from the top 10 ranking because of the downregulation, and other genes are joined the top 10 ranking because of the upregulation. If we look at the expression change rate, when under YEPD condition, the rate has no obvious change during 5 to 15 minutes, then has obvious upregulation on 30 minutes. When during KCI condition, there is an increase of expression rate during 5 to 15 minutes, and obvious upregulation on 30 minutes. From the biological process, we can know the expression of metabolism-related gene was downregulated. The expression of stress response-related gene was upregulated. SCOL1 has more obvious increases of expression rate across all the time points, but overall rate number is smaller than HOG1. For the biological processes, SCOL1 shows the same expression changes, but has more unchanged genes. Due to osmotic stress, the HOT1 gene is activated through the HOG pathway. The peak results shows the HOG1 and the SCOL1 have similar components. The expression rate changes also shows a similar trend of the regulation. After 5 minutes, the regulation of metabolism-related genes are downregulated, and the stress response genes are upregulated. From this result, we can know the genes that components the peaks was significantly changed. The regulation of gene SPI1 can show to encounter the stress, some unique functionality genes has been expressed. Gene GPD1 and FMP48 is shared between two datasets. The biological process of FMP48 shows chromosome segregation and protein phosphorylation. protein phosphorylation can happen by protein kinase such as HOG1, so this might show the signal transportation and interactions between HOG1 and SCO1. For the unclear questions, gene across all datasets may be because there are also internal or external stress under natural conditions. HOT1 YAPD has no peaks, maybe because of HOT1 is not activated. HOT1P gene thus appears in the RNA sequence data results. Gene STL1 shows in SCORE KCI result. Thanks for your listening.